For week 4, we will be covering the medically important eukaryotes. This is primarily coming from chapter 12. However, as we go more in depth with some of the medically important eukaryotes, it's very important for you all to read chapters 19 through 24 on the select microbial diseases that are caused by eukaryotes. That will be outlined in this particular lecture. This is a truncated summary of what you all will have for the next week. Your sub, Mrs. Felton, as well as Mrs. Sharistani, for the second lecture class, will be able to go over this more in depth. Again, if you all have any questions, you are to contact both of those individuals, and you can also contact me via email as well if any questions arise over this content. So again, in regards to week 4's lecture, it will be on characterizing, I should say, as well as classifying the eukaryotes, with a special focus on the medically important eukaryotic organisms. Now, when we're talking about eukaryotes, it's very important to note that because this is a microbiology class, we are only focusing on the microbial eukaryotes. However, we as humans are eukaryotes, so it's very important to remember that. Now, there are 5 major groups of eukaryotic organisms, and again, we're focusing on the microbial eukaryotes. They will be the protozoa, fungi, algae, water molds, and then slime molds. Within all of these groups, we do have those that are pathogenic, but then we also have those organisms that are vital or beneficial for human life. As you guys learned during week 3's lecture, we have various types of associations with these organisms and what we refer to as the micro-host relationships. So we do have symbiotic and synergistic relationships with certain types of eukaryotes, but we also have those parasitic and antagonistic relationships too. You all will learn more about the reproduction of eukaryotes. For now, what you need to know, however, is that it is more complicated in eukaryotic organisms than it is in prokaryotes. And that's due to the nature of the eukaryotic cell. So I would like for you all to go back and review chapter 3, which not only focuses on the structures of the prokaryotic cell, but it also focuses on the structures that are within the eukaryotic cell. In order to understand eukaryotic organisms and how they lead to disease, it is essential for you to read that chapter. So please make sure that you review chapter 3. Now, some of these organisms you already know of in detail. Of course, you know the algae. They are simple eukaryotic organisms. They are phototrophic, therefore we know that they go through photosynthesis, and their photosynthesis is considered to be oxygenic. They also use the same types of chlorophyll as plants and cyanobacteria. That would be chlorophyll A. Water molds are very similar to fungi, however they differ in the following ways in regards to some of the cellular structures. They have tubular cristae. Their cell walls are made up of different polysaccharides such as cellulose. And then they have spores that are flagellated. Lichens are actually partnerships of eukaryotic organisms. Typically, they consist of partnerships between fungi and algae as well as cyanobacteria. And these are abundant throughout the world. They usually are found growing on soil, rocks, and leaves, as well as tree bark. So if you've ever been out camping, you've probably come across this type of organism. They're very important in the creation of soil from rocks. They usually serve as a food source for many organisms. The protozoa, these are the eukaryotic microbes that you guys viewed during lab. All of these are free-living in water, in which case we said that they are trophozoite, which will be this term that you see here when they're actively metabolizing and living within water. However, when they are under adverse conditions, pretty similar to a bacterium, they can turn into a more resistant structure, and that resistant structure is known as a cyst. When they are in the cyst form, they can become pathogenic. Now, even though the cyst happens to be a resistant structure, very important to note here, cysts are not as resistant as endospores. So we can kill a cyst much more easily than we can an endospore. Similarly to endospores, however, cysts are not considered to be a reproductive structure. So protozoans, when they are under adverse environmental conditions, will turn into a cyst, and then when those environmental conditions have improved, that cyst will turn back into what we say is a trophozoite, which is the active stage. So some examples of pathogenic protozoans will be Entamoeba histolytica and Giardia lamblia. You will have a case study during week 4 on this particular organism, Entamoeba histolytica. Now, cysts can be shed through the fecal matter, and we can acquire them from infected individuals if we're handling their fecal matter and we don't wash our hands properly. So in that case, we are self-inoculating. Also, if an infected individual is handling our food and they're not washing their hands properly after handling their own fecal matter, they can also transmit it to our food as well. The next slide just goes more into the morphology of protozoa, a lot of which you've already learned in lab. We know that they have great morphologic diversity in regards to their cellular extensions. So you guys should recall that they have flagella, as well as cilia and pseudopodia that they use for motility. What you have here is some additional morphologic diversity in regards to the fact that they have 2 nuclei. And those nuclei are referred to as a macro and micronucleus. And they each have different functions that are listed here. Because protozoa lack cell walls, they have contractile vacuoles, which are a specific type of organelle that's responsible for regulating the amount of water that enters or exits the cell. Now this slide just goes more into microbial associations. I will modify this PowerPoint somewhat. So even though you had it in week 3, you may still see it in the week 4 lecture. So it's very important that you know this information. Now the last thing I'm going to discuss in this truncated lecture, because your subs will be going more over the disease states, is transmission of a pathogen. This is covered mostly in chapter 14 in your textbook. However, transmission is very important when we're talking about disease states because it is a very important part of the parasite's life cycle. So again, if we're having parasitic relationships with these organisms, and if it is a parasite and it causes disease, then we must acquire it some way. So transmission is very important. So to continue the life cycle, the pathogen and the parasite in this case must produce a stage which is capable of leaving the host. So I was just mentioning the fact that with cysts, they can be shed via the fecal matter. However, for most microorganisms, they are not viable for long outside of the host. So what that means is they are not able to survive for long periods of time when they are outside of a host. So they must produce some sort of structure such as a cyst, or in the case of bacteria, endospores, that will allow for them to survive for longer periods of time. So when it says here, capable of leaving the host, that means that the organism must be able to leave the host in some mechanism. So whether it's via the fecal route or whether it's via bleeding out from an injury, or if it's leaving via, say, the mucosal membrane. So if it's leaving via mucus. Next, it should be able to survive in an exterior environment. So again, it must be able to resist the outside environment, any conditions that may threaten its survival, so it can turn into a cyst. And then it must be capable of locating and establishing itself in a new host, in which case, again, this means that now, once it comes into us, whether it's via random contact or whether it's via we're eating food that has been contaminated, it must be able to now enter into the new host by a variety of means. Usually they're entering in via the open cavities in the body, such as the nasal passages or the oral cavity, as well as via the orbitals, in which case we usually self-inoculate ourselves. And at that point, it can begin to colonize us and establish itself within a new host. At that point, it will begin to grow and develop. So this is known as the maturation stage. Next, it will go through reproduction, in which case you have already learned in previous lectures that this reproduction can be sexual as well as asexual. And then there are various transmission phases that it has once it's within the host as well. So I'm going to actually end here with this brief introduction into chapter 12, which again specifically focuses on the eukaryotes. So please make sure that you all read chapter 12 as it gives you more information on the eukaryotes, but review chapter 3, particularly on the cell structure of eukaryotes as well. And then for your subs, Mrs. Fulton for the morning class, Mrs. Sharastani for the afternoon class. They will specifically be going over the various disease states of the eukaryotic microbes that I've selected for you all to learn. And then you will also have case study activities as well. Again, if any questions arise during this time, please feel free to contact myself or one of your two subs.